Hi everyone, this is the lecture on monomers and polymers for AP Bio, and your essential question is, how are large biological molecules built up from their smaller component parts? By the end of this lecture, you should be able to answer that question using the vocabulary here. So first, let's talk about how polymers are built up and how they're broken down, and then I'll give you a brief overview of the major classes of biological molecules, and their monomers. So first of all, you should know what a polymer and a monomer are, and if you need to pause the video to read the text on the slides, feel free to do so. When you're ready, you can resume the video. And you can do that at any time that you feel like you need to slow down this lecture. But basically, polymers are what you get when you take a bunch of monomers and you string them together. And the way that you string them together is what we call a dehydration reaction. Conversely, the way that you'd break apart a polymer is through a process called hydrolysis. And whether you put monomers together or whether you break them apart, you're going to need to use enzymes in order to do so. So here we have a graphic that shows you, how that happens. So here we have, this is a short polymer right here. It has 1, 2, 3 monomers. It has a hydroxyl on one side, and it has a hydrogen attached to this monomer on the other side. And so here we have an unlinked monomer, and it has a hydroxyl on one side and a hydrogen on the other side. Now when you put these two together, what's going to happen is this hydrogen is going to be attracted to this hydroxyl because of the electronegativity of the oxygen there. And so that's going to allow this hydroxyl and this hydrogen to basically bond with each other, forming a water molecule. And then what happens is this monomer here on the short polymer will then bond with this monomer here. As they lose a water molecule, they form bonds with each other. And so now you're going to end up with a longer polymer that now has 4 monomers on it. And this process, because you're losing a water molecule, it's called dehydration. It's also known as dehydration synthesis. The converse can also happen. So here we have a polymer that has 4 monomers, and if you add in a water, then what can happen is the hydroxyl, the OH from the water molecule, can add to one of the monomers, and the hydrogen can add to another monomer. This monomer happens to be part of the polymer, and it can break apart that bond right there. And so that's called hydrolysis. The prefix hydro meaning water, and the suffix lysis meaning break apart. Okay, so we know then that you can take monomers and link them together to make polymers. And by the way, this reaction, this hydrolysis reaction, would be catalyzed by an enzyme. And this dehydration synthesis reaction would be also catalyzed by some other enzyme. But there are lots and lots of different polymers that you can get from different collections of monomers. And then because you can have different monomers linking in different ways to form these polymers, the polymers will have different structures, and that gives them different functions. And that means that these polymers can carry out all of the functions that living things need to be able to do in order to stay alive and maintain homeostasis. So here I'm just going to give you a brief overview of the 4 major classes of biological polymers. We also call them biological molecules or macromolecules. So the first major class are carbohydrates. Their monomers are monosaccharides, and they look like this. They basically have a ring of carbon. Sometimes it's a chain of carbon, but we often see it in the ring form in biology. And everywhere that you see a vertex on this shape, there's an invisible carbon there. Similarly, you can see that you have hydrogens and hydroxyls at each carbon, like almost every single carbon here. And so this monosaccharide is the monomer or the basic unit for all of the carbohydrates that make up your cells and that you eat and that you use for life. If you take two monosaccharides and put them together, you get a disaccharide. And if you have three or more, then you would have polysaccharides. And these are the different types of polysaccharides. Here you can see their functions. There's going to be a separate lecture just on carbohydrates, but it's a good idea for you to pause the video and just read through the different functions that you'll get with these carbohydrates as a way to prime yourself for the upcoming lecture on carbs. The next class are lipids. So lipids are all hydrophobic, meaning they hate water. And the components, fats are made of triglycerides, also called triacylglycerols. Here's a simple model for that. Another type of lipids are phospholipids, which make up cell membranes, and here you can see a simple model of a phospholipid. And then the last type of lipid are steroids, and you can see on the bottom, a steroid basically is a carbon and hydrogen structure that's made of fused rings. And you can look at the functions of lipids, but there is going to be a separate lecture just on that. Next are the proteins, and the monomers of proteins are amino acids. They have a carbon in the center, they have an amino group on one side, and they have a carboxylic acid group on the other side. So they're called amino acids. And then they'll have some variable R chains. So that just means that there could be different things that are attached to that central carbon here. And those different things will give different amino acids their properties. Here are some examples of the types of proteins you get when you put amino acids together, and here you can see their functions. And of course, there's a lecture just on proteins coming up for you. Last of all, the last major class of polymers are nucleic acids, and there are two main types of nucleic acids, DNA and RNA. But for both of them, their monomer is the nucleotide. It has a pentose sugar, pentose meaning five-sided. It has a phosphate group, and it has a nitrogenous base. And the five nitrogenous bases are C, G, A, T, and U. Their functions are over here, but primarily they basically store hereditary information that's used to make proteins. And there's a separate lecture just on nucleic acids. Here are your key takeaways. So monomers link together through dehydration synthesis to form polymers. Polymers break down into monomers through a process called hydrolysis. And remember, both dehydration synthesis and hydrolysis are catalyzed by enzymes, which are, of course, a type of protein. Macromolecules are very diverse in their structure, which makes them diverse in their function, and they fall into these four major classes: carbohydrates, which are built from the monomer monosaccharides; lipids, which have different types of components; proteins, which are built from the monomer amino acids; and nucleic acids, which are built from the monomer nucleotides. And last of all, just remember the term macromolecule is basically the same thing as the term polymer when it comes to biology. Okay, I'd like you to just take a look again at the essential question. And if you can answer this question using all of the vocabulary shown here, that means that you are ready to move on to the individual lectures on the four major classes of polymers in biology.